Assalamu alaikum students, this is the second lecture of botany and this second lecture is related to the plant cell wall. This is a portion of the anatomy portion of your syllabus and today we are going to study the plant cell wall with the help of the diagrams and the given slides. The plant cell wall is found in plants, algae, fungi, bacteria and some archaea. Animals and protozoans do not have cell walls. In plant, cell wall is located outside the cell membrane and provide these cells with structural support and protection and also acts as a filtering mechanism. हमें पता है कि हमारे पास सेल की जो आउटर मोस्ट बाउंड्री होती है इन केस ऑफ एनिमल सेल्स दैट इज द सेल मेम्ब्रेन. सेल मेम्ब्रेन जो होती है वो डिफरेंशियली परमिएबल होती है, लेकिन प्लांट सेल वॉल जो है वो सेल मेम्ब्रेन के बाहर होती है और ये सेमी परमिएबल नहीं होती, रादर ये पूरी तरह चीजों को अंदर आने देती है, but it acts like a filter. So we will go into the details of what we mean by it acts like a filter, but what we need to understand at this stage is that almost all the plant cells do have it, with the exception of protozoans and animals. All of the plant cells are having it. Now coming to the next slide. What constitutes the cell wall? The important components is termed as chitin. You are very much aware of the term chitin. It forms the microfibrillar component of the cell wall of most fungi and is principal components of the hard exoskeleton of many invertebrates as well. Chitin is present in some lower invertebrates as well, and it also forms an important microfibrillar component of the cell walls of fungi. It is having certain structures which are bonded together with the hydrogen, and these chitin molecules are formed with the help of the hydrogen bonding. Second thing that forms the cell wall is termed as the cellulose. Cellulose is the significant structural polymer in the primary cell walls, while chitin is the main structural polymer found in the fungal cell wall. So what we need to understand at this stage is that fungus is having chitin, whereas cellulose is forming the cell wall of most of the higher plant cells. Another component of the cell wall is the lignans. Lignans are high molecular weight insoluble plant polymers, and they have complex and variable structures. Lignification of the cell wall occurs after the laying down of the polysaccharide components of the wall and towards the end of the growing period of the cell. The purpose of lignification is to strengthen the cell wall by forming a rigid structure throughout the matrix of the cell wall. So lignans are the structures which give strength to the cell wall, and these structures along with the chitin are present in case of lower plants and some type of fungi in which they are present. Proteins are also forming an important structure. The constituents of proteins or different type of proteins, they also form an important component of the cell wall. There are two types of proteins we observe in the cell wall. One is termed as the enzymes and the other one are the structural proteins. The enzymes, we are very much aware of them. They are ATPases, DNases and RNases and certain phosphatases. They are present in the cell wall, and when the process of making some of the molecules enters into the cell wall takes place, they bind themselves to these molecules and make the passage of these molecules easy while entering inside the cell membrane. And similarly, there are structural proteins. These structural proteins are concerned with the cell wall extension. Water also forms an important structural component of the cell wall. It forms part of the gel structure of the pectins. Because of the changes in the water content of the wall causes reversible changes in the texture of the cell wall matrix as the pectin gel changes to a viscous solution. Water plays a very important role because it makes the cell membrane permeable, cell membrane and cell wall permeable to different molecules that enters into it. Similarly, the growth of the cell seizes the space occupied by water in the wall, becomes progressively filled with lignin, thus making the matrix and therefore the whole wall much more rigid. There are some incrustating substances which are present inside the cell wall. Incrustating का मतलब होता है कि वो चीजें जो cell wall को strengthen करती हैं, वो cell walls को बिल्कुल इसी तरह जिस तरह आपके पास हम कोई दीवार बनाते हैं और उस दीवार के अंदर हम फिर सीमेंट लगाते हैं, and that cement is actually meant to strengthen the wall, or we put some sort of iron rods inside the cell wall. Similarly, these substances are meant to give strength to the cell wall, and these substances, which are termed as the incrustating substances, they are few different types of them. One important amongst them is the chitin and the suberin. The functions of these chitin and suberin are given in that slide, and you go through it and you will find out what is given in this slide, that is explaining the role of the chitin and the suberin which is present in the cell wall, and they makes the cell wall more hard, more rigid, and more it provides more strength to the cell wall. Now have a look at this diagram. This diagram is showing the different layers which are forming the cell wall. The outermost layer is chitin with intracuticular wax. Then we are having the pectin. You are very much aware of the term pectin, because when we were studying cell wall in FSC class, we were aware and we learned that term, the pectin, the lignin, the suberins, they are all part or important constituents of the cell wall. Similarly, you can see that there is the chitin which is forming the intracuticular wax, then the pectin, the cell wall, and the plasma membrane is present over there. This diagram is explaining the basic layers which are formed in case of the cell wall. Now have a look at this. This diagram explains that when a cell becomes more specialized for performing its different roles, like the xylem cells, we have got certain depositions of more materials towards the inner side of the cell, and they form the secondary and the tertiary layers. This diagram explains how the inner layers are developed inside the primary cell wall of a cell, and these cells makes the cell wall very much thickened. We are having a specialized layer which is termed as the middle lamellae. Middle lamellae bind the two different cells together. Then we are having the primary wall, then the secondary wall, then the tertiary wall, and these walls give thickness to the cell wall of the different cells, especially those cells which have to perform very important functions like the xylem cells. Now, secondary wall is often deposited on the inner side of the primary wall after the growth of cells stops. It is 4 to 10 micrometer thick, rigid, non-elastic, permeable, and made up of cellulose and lignin deposits. Secondary wall deposition is not uniform. At some places, secondary wall is not laid down. Such unthickened areas are called pits. Pits of two neighboring cells form pit pairs. While we were studying cell wall, we came to know, and I explained it, that when the primary wall is laid, afterwards the secondary wall is laid towards the inner side of the primary wall. This secondary wall is not uniformly deposited. At some areas ये अपने दरमियान में छोटे छोटे से सुराख छोड़ देती है. ये जो सुराख होते हैं ये पोर्स कहलाते हैं, या हम इनको पिट्स कहते हैं. जब आप लोगों ने अपना एफएससी का लेक्चर देखा था तो उसके अंदर हमने देखा था कि छोटे छोटे से पोर्स जो हैं वो हमें जाइलम सेल्स के ऊपर नजर आते हैं. ये जो पोर्स होते हैं इनको कि हम पिट्स कहते हैं. पिट्स जो हैं वो हमारे पास एक किस्म की नहीं होती, बल्कि ये 2-3 किस्मों की होती हैं. इनके ऊपर हमारे पास बॉर्डर्स भी हो सकते हैं, ये बिल्कुल सिंपल भी हो सकती हैं. मैं आपको डायग्राम में दिखाऊंगी जो अगली स्लाइड पर है कि हमारे पास पिट्स जो हैं वो कैसे बनती हैं और पिट्स के गिर्द हमारे पास किस तरह की डेपोजिशन होती है जो उनको पिट पेयर्स बनाती है. So this is the diagram that is showing the pits. Have a look at it. We are having a certain opening, and this opening is termed as the pit, which is like a hole or an opening, and it has a certain deposition in the middle. This is termed as the torus. We are having the pit aperture, then the pit border, and the pit membrane. I will be providing you some notes that will be showing you pits in more detail. Presently, you need to understand that secondary walls are not equally developed, rather it leaves some areas. These areas are unthickened, or they are very slightly thickened, and these areas form the pits. These pits can be simple, these pits can be bordered. So in this diagram we can observe that what kind of pits are present in xylem cells. They can be of different types. Now you can see with the help of this slide that simple cells, that is the parenchymatous cells, are only having the primary walls, whereas the specialized cells, they are having the thickened walls, and these thickened walls we observe in case of the xylem and the phloem cells, in case of the fibers, and they give strength to the plant cell because they have to make them rigid, and they give them the strength that is required by the plant to stand against the gravity. Now this is the structure, or we can say the ultrastructure of the cell wall. You can see that there are certain specialized long structures which we term as the microfibrils, which are shown in green, and then we are having the hemicelluloses. They are shown in green, dark green, and then we are having the pectin substances. They are the gelling matrix. Then we are having the glycoproteins, and these structures, what they do is that they form a very stronger structure which forms the cell wall, and we can call that this structure is termed as the ultrastructure of the cell wall. So this is also explaining the at a very lower or at a very finer level. If we study the cell wall, we will find out that it is form of long strands. First of all, they are termed as the macrofibrils, then is the microfibrils, then we have the micelles, then the cellulose chains, and then the glucose molecules. They are forming the basic constituents of the cell wall. This is how the structure of the cell wall is formed. We studied the chemical composition, we studied the structure of the cell wall, and here we go with the structure. Now, how cell wall divides itself? At the point when the cytokinesis takes place, means when the cytoplasm divides, we are having specialized vesicles which are formed by the Golgi apparatus and the endoplasmic reticulum, and these small vesicular structures, they are built in the center of a dividing cell, as you can see from the diagram, and when they come close to each other, they form the cell plate, and this cell plate divide the cell into two portions, and the two cells, when they divide, then with the help of the incrustating substances and with the help of the water, they divide the cell, one cell, into two, and then the cell wall enlarges, it stretches itself and it becomes larger. You can see that this diagram, it's showing that how a single cell is dividing into two plant cells, and each is having its own cell wall with the help of the phragmoplast that forms the cell plate in the middle of the cell. So if we say what are the functions of the cell wall, they are very simple. The major role of the cell wall is to form a framework for the cell to prevent overexpansion. It provides mechanical strength. It bestows definite shape. It protects the cell from mechanical injury. Cell communication with one another with the help of the plasmodesmata. You are very well aware of the term plasmodesmata. We have studied it a number of times during our first semester. Plasmodesmata, as we know, the cytoplasmic strands, and it provides a porous material for circulation, for the movement of water, minerals, and other nutrients. Cuticle present on the outer surface of the epidermis cell leaves and suberin present in the periderm prevents water loss. Cell wall has an important function to regulate how plant cells achieve their final size. Similarly, some of the proteins of the cell wall possess catalytic activity by acting as enzymes. So girls, this is the last slide of the lecture, and in it we will conclude the properties of the cell wall. The cell wall provides shape and support to the cell, and it appears to be rigid. In some plant cells, a secondary wall is developed after cell ceases to grow. Additional layers may be formed containing lignin in xylem cell walls or suberin in the cork cell walls. So, concludingly, we can say that cell wall is a very important structure in case of the plant cells and fungal cells and bacterial cells. It possesses different properties. It has some structural specialities. It is absent in animals, and it grows and it performs very important functions in case of the plant cells. So that is the end of the lecture today. Thank you very much. I will be providing you notes that will be support you to understand this topic. You can use these notes, you can use these slides, and I hope this topic will be easily understood by you. Thank you very much. I'm Rahila Zafar from Botany Department. Thank you.